So today, like I said, I want to continue talking about peripheral B-cell responses, and then start our discussion of NK cell responses. And this is where we left off on Wednesday. So remember that we were kind of talking about activation of B cells in the periphery. We saw our B cell that had come out of the bone marrow as sort of a naive B cell, interacting with antigen and sort of starting this activation process, but then needing helper T cells and other kinds of stimuli to start expansion and differentiation that can include class switch and affinity maturation, and all of this was taking place in the germinal center as a part of the germinal center reaction. And so we talked through that whole process and sort of all of this big picture last time. I talked through kind of the general idea of isotype switching or class switching and of somatic hypermutation, where we get mutations in the variable region of the antibody gene so that the affinity of the antibody changed, and so that we then got that affinity maturation happening of the protein. In both of those cases, I sort of mentioned that we need to have a change to the DNA happening of the antibody encoding gene. And so again, if you remember, we had antibody gene changes that were happening during B cell development in the bone marrow with the enzymes RAG1 and RAG2, and then I told you we're done with RAG1 and RAG2. But in fact, we're not done with changes to the DNA. So now we're in a different location, different time in the B cell's life, but we are again going to change the DNA. And like I said, when I finished undergrad, I sort of learned basically what you saw on this slide. And we kind of talked about, and there are changes to the DNA. And sort of right near the beginning of the time when I was in graduate school, we actually learned about the enzyme, the enzymatic machinery that allowed for class switch and affinity maturation to happen. One thing that's really important to know is that we're going to see an enzyme that's going to be changing DNA here, and it's not RAG, it's a different enzyme. Again, we're a different location, different time in the B cell's life, different enzyme that's going to be changing the DNA. One of the things that I think that immunologists learned, and at least I as a student was surprised by, I don't know if like the people who like were really in the know at the time were surprised, but I was surprised, was that it turns out it's actually the same enzyme that's doing both of these things. Even though these seem like really different changes to the DNA, it's the same enzyme. And so people are still kind of arguing about, we know the enzyme gets turned on in the germinal center, but like the timing of when it's doing which one is still kind of murky. All I will say is both of them magically in the germinal center with this enzyme. And one of the things that people knew for a long time, and this was particularly important when they were thinking about affinity maturation. If you remember, I told you that affinity maturation and somatic hypermutation involved point mutations. So we were getting single bases getting changed to other bases. One thing that people had sort of noticed is that it wasn't just any old base that seemed to get changed. We were always seeing changes in bases in the heavy chain and light chain variable regions, but it was also C's that were getting changed. So whatever thing got changed, it was a C at the beginning and got changed to something else. I will also point out, and I will say more about this as I go forward, when we now know that when class switching happens, when we have isotype switching, that I pointed out the switch regions or the switch sites that were in front of the constant region genes. And I told you that sort of DNA changes at the switch sites were pretty important in this process. People also had observed that the switch sites had a lot of Cs. So there's something going on with the C base pair in both of these processes. And what we ended up finding was that there is a specific enzyme. That enzyme is known as AID, that leads to both the processes of somatic hypermutation and class switching. AID is only made in a B cell when that B cell is doing its activation process. So it is activation induced. And oh my gosh, the AI in AID stands for activation induced. AID is an enzyme that catalyzes a specific reaction. That reaction is a reaction that happens on cytidine or C in the DNA. So remember I told you that cytidines or Cs were kind of important in this process. And you can see the structure of a cytidine, a C, right here. And just so you are sort of aware or sort of thinking about this, this part up here is involved in the base pairing. And in the case of a C, what's it supposed to base pair with? G, right? This enzyme, activation-induced enzyme, chemically modifies cytidines, or Cs, and takes this part, where you see an NH2, and changes it to an oxygen with a double bond, like you can see here. And so you can see that change chemically being made.
That's the portion that is involved
in base pairing, yeah. And so you can see kind of the before and the after molecule. We're removing this NH2 group from cytidine, and so we call this a cytidine deaminase because it removed this amino group. So this enzyme is known as activation-induced cytidine deaminase. Activation-induced because it's only made in the B cells when they're activated, and it removes an amine group from cytidine. So it's activation-induced cytidine deaminase. Ideally, you can imagine immunologists might have wanted to call this AICD, but that abbreviation was already taken when this enzyme got described. So instead, the enzyme got named AID, activation-induced deaminase, even though it's specifically deaminating cytidine. And the key thing that you want to think about when you think about this cytidine deamination reaction that you're seeing here is that, again, at the beginning we had a C base pair in the DNA. When this chemical change is made, this new part, you can see already that the change that was made was in that part I said was involved in base pairing. This product is actually uridine. It's uracil. It's U that you learned about in Bio 250. So this enzyme changes a C to a U. Why is it what's important? Why is the cell perhaps maybe going to notice that we had a change of a C to a U? And again, we changed the base pairing part, so that's part of the whole change C to U. What is up with, why is this like a thing the cell should care about? Yeah, David.
Our DNA doesn't contain U's?
Yeah, first of all, this is a reaction that's happening to DNA. U is only in RNA in our cells. And we just put a U in the DNA. So, this is wrong. Right? Anything else that's probably wrong? What is it?
The base pair match, G will match it.
Yeah, it's like a cross from a G. Now you basically turned what was a CG base pair into a UG base pair. And U doesn't go with G. And we're in DNA, and we got U in DNA, which we're not supposed to. This is actually a bit of a problem for the cell. And so the cell is basically now getting this U in its DNA where it shouldn't have a U. And the cell has to deal with that U. It has to resolve the uracil. So basically what you can see is here's AID. There was originally this CG base pair, and AID changed it to be a UG base pair. Sometimes the cell doesn't notice. So first let's imagine that the cell doesn't notice. We've got two pieces of DNA. They're going to separate, and they're going to make two progeny pieces of DNA. One of them is going to be GA, right? And when that one is GA, what's going to happen when we try to replicate that GA? Yeah, I don't know. It'll be CT. It'll basically make what we had in the beginning. There's actually no change to this piece of DNA. But the other piece has UT. So what's going to happen when we try to replicate that UT? Yeah, David.
The U is going to pair with A,
Mm-hmm.
and the T is going to be the same.
Yeah, so where we originally had a CT, we're now going to turn it into a TT. So we're going to get a point mutation in this piece of DNA. So if the cell doesn't actually ever notice the U, then we're eventually going to get a one base pair point mutation where we're going to move a C to being a T. Okay? That's a point mutation. That can be one of the mutations that we might see for this somatic hypermutation process. Alternatively, the cell might notice the U and might try to use DNA repair processes for that U. And one of the things that it could do is it could cut out that U. And so here you can see it cut out the U and left the rest of the DNA, like left the backbone. And that might be something that could happen for like one single U. When that happens, a type of DNA repair occurs where basically any base pair gets put in. It could be any of the four, just kind of randomly the polymerase puts one in. So again, that's going to give us a point mutation. It's going to give us anything, which is going to pair with whatever. So those are two examples of how putting a U in the DNA can lead to a point mutation here. And so one thing that will happen is, again, we'll have AID leading to these point mutations. And AID will specifically attack cytosines in the variable region of the heavy chain and the light chain gene. That's the only place that AID can really attack and change C's into U's. And so as a result, we're going to get a bunch of point mutations at that location. That is going to change the affinity of the antibody. And so this cytosine or cytidine deamination is the process that is actually going to give us the point mutations of somatic hypermutation. And so that's how B cell is going to actually be able to do that affinity maturation as a result of somatic hypermutation. Yep, Brenna? You have a question?
I just don't know why the cell is doing this.
So Brenna's question is, why is the cell doing this? What does it give the cell? What's the benefit to the cell of doing this?
Just a variety of antibodies.
So it's going to give you a variety of antibodies, and it's also going to make those antibodies bind to antigen better. And so we're now going to have a better, improved response to the antigen over time. And so the cell is doing this to improve antigen. And it's doing it really to improve antigen recognition for the organism. It's possible that the cell might destroy its own personal receptor, but somebody in the population is going to improve, and so the organism's going to be better off because all the B cells are doing this. Somebody's going to win the lottery. Does that make sense? As I mentioned, there's sort of one other way that DNA repair can happen with these uracils. This DNA repair is what happens really if there were a bunch of C's next to each other, and so we had to repair like a whole bunch of U's next to each other. So instead of if there was just one C that got changed, and so we put in one U, imagine we had like a bunch of U's all of a sudden. Like this would be a bit of an emergency for the cell. The cell would be like, oh crap. In that case, we're actually going to cut out a whole big region of the DNA. We're going to have a double-strand break. And the cell is going to go through a different kind of DNA repair. The way that the cell basically does this DNA repair is that I'm going to like personify the cell a lot here. The cell does not actually have any wants, but we'll pretend it has wants because it's easier to phrase. The cell is like, oh crap, I got this like totally ruined piece of DNA. I don't know how to fix it. And the way the cell is actually going to fix it is it's going to look for a similar piece of DNA elsewhere in the chromosome and recombine. It's going to sort of try to find a piece of DNA that sort of matches and that is intact and smush the two together. Basically using the intact one as a template. This is called recombinational repair. I know that Dunaway mentions it really briefly in Bio 250, but you may not remember it. In fact, the only reason I know that he mentions it is because once upon a time he was sick for the DNA repair lecture and I had to give it off of his slides, so I know there's a slide.
Recombinational repairs.
Yeah. And so like I said, basically what the cell does is it tries to find another piece of DNA that has the same sequence to sort of use for repairing by joining them together. If you recall, with class switch, in front of each of our different constant region sections, there's a switch region. And the switch region DNA is the same exact sequence. So this is identical sequence to this, and it's identical sequence to this, and it's identical sequence to this. All the S's have identical sequences. And that identical sequence has a lot of C's in it. And so what will happen for class switch is that AID will basically destroy a switch region, and the cell will do a recombination event with another switch region to try to repair. And so you can see we might join together two switch regions, and the DNA that was between those two switch regions will get eliminated. And so there are specific reasons why the cell might say, oh, I'm gonna destroy the IgM switch region, and the only one that's gonna be available is IgG2b, and so thus I'm gonna switch to IgG2b. So there's sort of this idea of which switch region is available. That's based on which cytokine the cell has received. That will make the cell switch to different isotypes. But what I hope that you see here are the ways that this one enzyme, AID, can lead to these two important processes that happen in activated B cells. This one enzyme gets turned on during the germinal center reaction, and that leads to both of these processes happening to the B cell. One other thing that I want to briefly mention is that there are patients who do not have AID, so they are missing AID. And these patients have a specific phenotype. They have a specific disease. And so I'm going to show you on the next slide details of that specific disease. It turns out that this disease was actually known about before we knew about AID, because in reality, there are 3 different genes that can be mutated that all give you this same disease. AID is one of them, but there are 2 others. So if you imagine for a second, someone who doesn't have AID, what do you think you would observe in a patient who doesn't have AID? What would be there? What would not be there? Yeah, Brenna.
I would think autoimmune immune deficiencies because
Okay.
they weren't able to make the correct isotypes
Okay.
needed for the specific.
So they're not going to be able to class switch and make the correct isotypes. Are they going to have any B cells at all? Yeah, they're going to have B cells. Those B cells are going to be able to sort of do development. They're just not going to be able to get activated later in class switch. Are they going to have any antibodies at all? They are going to have antibodies. What kind of antibodies are they going to have? IgM. They're going to have IgM, and they're only going to have IgM. So based on percents, they're going to have 100% IgM. So what might you say is one of the big things you're going to see in these patients? I basically said it, but you can rephrase it to me. Yeah.
Like lower affinity antibodies?
So they'll probably have lower affinity antibodies, and they're going to have too much IgM. And that's really because they have too little of everything else, right? It turns out there's one other thing that these patients have. And that is, these patients don't actually have germinal centers. It turns out you can't do this whole germinal center process if you don't have AID. And so these patients have a disease that's known as hyper-IgM syndrome. It's really because the first observation was you have too much IgM. Well, yeah, the reason why you have too much IgM is because you can't make the other ones. You also have no germinal centers. So this is actually a lymph node in a normal individual. You can see lots of germinal centers. This is a lymph node of a person with hyper-IgM. No germinal centers formed whatsoever. So we get no class switch. We get all antibodies are IgM. You can imagine we're going to have some immunodeficiency, exactly as Brenna said. We're going to have low affinity antibodies, exactly as Juliana said. This slide also mentions one of the other genes that could be mutated. Because if you have an AID mutation, you have hyper-IgM. The other 2 genes that can be mutated for this are CD40 ligand and CD40. If you recall, when our B cell is getting activated, it has to get help from a T cell, and that T cell is providing some help in the form of a cytokine, but also in a protein-protein interaction with CD40 on the B cell, interacting with CD40 ligand on the T cell. We said that was kind of like CD28 and B7. If you're missing either of those proteins, again, you have B cells, the B cells just can't get activated, can't class switch, can't make a germinal center, all that stuff. And so that also leads to hyper-IgM. Since I switched around the order of topics, I kind of already did a kinetics and memory day. So there are a couple of points about kinetics and memory B cells and antibodies that I want to make. I showed you this slide before about primary versus secondary immune responses, and I largely was focusing on sort of T cell issues, but we see some B cell changes as well. So again, at the primary response, we've got a small number of B cells that respond at the start. We have more after they've gone through expansion and through a response. There was a delay in the primary response before we get the antibodies made. We're going to be making those antibodies right away, and in fact, we still might have some left over from previous infections in the secondary response. In a primary response, our response is all IgM related antibodies. and they have kind of a mixture of affinity, so they're not great affinity. Later on, we have class-switched antibodies, so we have antibodies that are going to be better adapted for dealing with a pathogen, and they're going to be high affinity. And so this is a big part, Brenna, of what the B cell's getting from doing this process. It's going to be easier to activate these cells, largely because they are effector cells, and we're going to see that cooperation between innate and adaptive immune responses. And so you can kind of see this here. So notice that during the primary response, we've got this lag, and then we do start to make some antibodies. This black line with like white under it is all antibodies, and at the beginning, it's all IgM. As time goes on, we get stuff that's not IgM. Most of it's IgG. Some of it can be some other stuff too. IgG is the most common, so mostly it's IgG. Now you can see if we get infected again, we're going to get a secondary response. It's going to be way bigger, and that's mostly going to be dominated by IgG because the cells that are responding have already gone through the germinal center. They've already class switched. We will get a little bit of IgM from some naive cells that are responding, but it's really predominantly an IgG response. And so here you can see this from your textbook as well. At the beginning, it's IgM in green. Over time, IgG kind of takes over. And with later infections, IgG is really the one that's increasing and is not increasing too much. Here you can actually see binding strength. So at the beginning, we got some IgM with kind of meh binding strength. Eventually, binding strength is going to go up, but it's really going up with the IgG, with the B cells that have already gone through the germinal center reaction. The M's aren't really changing too very much. And this is just the other way that your textbook kind of shows this. So you can see that in the primary response, the B cell responding to whatever antigen is sort of rare. It's more frequent in the secondary response. Especially the secondary response is really just going to be our class switched antibodies, G, A, and E, high affinity, lots of mutation. Also shown here. And there's again, a lot of this is the same things you've seen on the previous couple of slides about the peak being smaller or larger, IgM versus other isotypes, lower affinity, higher affinity. But the one last reason why I put this table in as well is because of the last line. Remember that for our B cell responses, to do this germinal center reaction, we needed to have the B cell get some help from a T cell. If you remember when we talked about antibodies and the antigens that they recognize, antibodies can recognize DNA, carbohydrates, proteins, and lipids. And when we talked about T cells, we only saw T cells that were responding to protein antigens on MHC. And so you can make a primary response really to anything, but if you want your B cell to get T cell help, the antigen has to be a protein, because the only thing a B cell can show a T cell to ask that T cell for help is a protein. And so this is also a big difference in some of these responses. Again, when we make vaccines and we try to take advantage of this, sometimes there are some vaccines where we would really like you to make a lot of antibodies to a carbohydrate. The most famous vaccine I'm thinking of is a carbohydrate antigen. And it's like, well, crap, how do we get you to make huge response that's IgG and high? Like, how do we do that when it's a carbohydrate? The answer is actually that we do a bunch of biochemistry and we glue a carbohydrate and a protein together. And so then there is some protein and some carbohydrate, and you can get the antibody made because you're linking those two things together. So this actually ends up being a really key thing, and it ties in with what we've talked about of the different types of antigens that the two cells can see. The other piece that I want to sort of mention here is that when we think about sort of kinetics and memory responses, I think that sometimes when I say, well, read a press article about the immune system, they'll say the immune response worked or didn't work. Like, it's basically the only choices of the immune system are on or off, right? Those are the only choices. But in reality, I hope you realize that there are a lot more variations between on and off in the immune response. And so sometimes if someone says, well, do I have an immune response? I have to be like, what kind? Give me a lot more information, because there's no just on or off. And so over time, following an infection, the kind of immune response you have might change. The types of protection you have might change. And so to give you an example, so let's imagine that you have an infection and we see a nice primary response. That primary response, at the beginning, you're going to have a lot of antibodies. Like if we're, say, here, you got a lot of antibodies. The microbe is gone, but you still have lots of antibodies. You are probably going to have enough antibodies to block transmission. You're never going to get that microbe again. Later on, you're going to have less antibodies, but maybe you have better memory cells because you had enough time for memory cells to develop. And so now maybe you have a lot of memory cells, and maybe those memory cells, so see, at the beginning, maybe you have mostly just antibodies. Maybe later you only have things that are circulating, or maybe there are sort of just the resident cells that live in your nose versus ones that live in your blood versus ones that live in your lymph node. And so what kind of protection, and I specifically have mentioned this before, in terms of are you protected from infection or are you protected from disease, might change over time. The data that I've seen, for example, on COVID vaccines is that after you get a COVID vaccine, your antibody levels are so high that you are completely protected from all infection with SARS-CoV-2 for about 7 weeks. But after that, you're protected by memory cells. The idea being that they trigger quicker. So you might get infected, but you might get rid of that infection really easily and not get very sick. And from what we can tell, we haven't found how long those memory cells last yet. As in, we've only been doing this for 4 years, and they totally last 4 years, and we think they last a lot longer than that, but we gotta wait and see. But the other thing is only 7 weeks, and so note that that protection can vary over time. One other thing that I want to mention is, as I told you before, at least for me, some of the details of germinal centers, the germinal center reaction, and things like that are places that we as immunologists have learned a lot of immunology during the pandemic. There are so many cool papers that talk about so many cool things that we have learned about this process during the pandemic. This was actually why I didn't pick up paper for next week until at the very last second, because there are so many cool ones about this stuff that I had to be like, what am I going to do? Which of them am I going to pick? And I picked one that's more about T cells, and I love that paper, so that's why. But I will tell you, there's a lot of really cool stuff. To give you one example, some people have actually started to measure how long they can actually track the germinal center that happens following certain vaccines now, and actually see how long does that germinal center last and what goes on, and do new cells invade and all sorts of things like that. One of the papers on that is a paper that I cite when people ask me, when I go get 2 doses of a vaccine, should I get them in the same arm or different arms? And my answer right now is same arm, because the germinal center might still be active. And so you want to re-hit that same germinal center instead of hitting 2 different germinal centers, because there's a different germinal center in different lymph nodes for each arm. And so there are actually 2 different papers people use. One says you should do same arm, one says you should do different arm, whatever. I like the same arm paper. But that kind of stuff we have actually been able to start to really do in terms of measuring germinal centers and understanding germinal centers. There's so many cool papers. But I do want to show you one sort of piece of data that I will tell you, like, I get it as a biologist now, but when I first saw this and had to think about what was going on here, it absolutely blew my mind. And so it's so cool. And it's so cool as like a big picture biology thing. So we talked about the germinal center reaction. And I described the germinal center reaction as sort of being like Darwinian evolution on the level of a cell in one little compartment of your body. In one little lymph node, you are actually getting a population of cells undergoing survival of the fittest to see who can survive to get antigen and get their pat on the head signal after they've made mutations. And if we think about this in terms of big picture evolutionary biology, we can be like, yeah, that's how organisms like get better. It's like also how we get high affinity antibodies, right? But you can also realize that sometimes evolutionary biologists talk about this as like, this is how we get really good variation. This is how we get some mutation and then we get specialists for different things, right? So these are this is some data looking at antibodies against SARS-CoV-2. In both of these situations, the authors were looking at antibodies to the original strain of SARS-CoV-2, not any of the variants, before mutations, like the very first version of the virus. And one thing that these authors were able to show is that over time, if we looked at the concentration of antibodies that was effective, basically how good were the antibodies got better over time because they went through somatic hypermutation and they could bind stronger. So they needed less antibody to bind. So you can see over time, antibodies got stronger at binding to their antigen. Yes, that's what I told you should happen in a germinal center. And here you can again look at antibody amounts. This one is actually showing a vaccine setting. So, after you get your second dose, your antibodies are at a particular level. They go down over time. You get another dose of vaccine. They go back up. You get more antibodies. Like, all this makes sense, right? This is against that original antigen you saw. Great. But the thing that most people worry about is that as time goes on, and as time has gone on with SARS-CoV-2, and with any virus, this would be the case, we start to see the virus mutating. And so when you wait for some number of months or things, it's unlikely that you're going to get infected with the same virus. The virus is likely going to mutate, right? And so it's not going to be the same antigen anymore. It's going to be a closely related antigen, but not necessarily the same antigen. And so part of the idea has been, well, this is cool and all. My antibodies got really specialized over time, but did they get too specialized? Did they now bind to original virus so well that they can't bind to new virus, and so now I'm out of luck? Do you understand sort of the problem? And so this actually, in practical terms, gets into this question of if you get another dose of your vaccine, does it need to change? Do you need to get a booster against the new virus now? Because the virus has changed. You don't want antibodies against old virus unless you are planning to use your time machine and travel in the past. You want antibodies against new virus. This group did this amazing study where they actually looked at how antibodies in germinal centers change over time to mutant viruses. And so these Beta, Delta, and Omicron are viruses that were not present and were not in the vaccine. But in fact, as a result, and so you can see at the beginning, Beta in particular, you have pretty low amounts of antibodies. While to the original strain that you actually got vaccinated against, you have better antibodies. So at the beginning, yeah, your vaccine only protected you against the strain you got, the Wuhan strain, not against the variants Beta, really not against Omicron, and against Delta. But it looks like if you actually let a germinal center reaction go on and you let this mutation process happen and you let natural selection happen, you get antibodies that are broader and can actually respond to more variants as well. And so it's not just natural selection for like being super specialized to this one antigen. It kind of ends up being a nice specialization to similar to mutant or similar family members. You get this little broadening. Like I said, I personally did not expect this. I don't think a lot of people expected this. And it has led some people to say, do we even need to change the vaccine? Can we just keep giving you the same vaccine over and over again and just make sure this happens? Because in fact, it's not clear if changing the antigen even makes a difference at all at this point. If you just got the same original vaccine over and over again, we think from the data we've seen that this broadening happens. And because of the germinal center, you pick up mutations and you actually get a little bit of broadening of responses. And so I'm showing you this more as a way that like immunologists right now are learning so much about this because of sort of the fancy techniques we have and also the massive number of people who all are experiencing the same antigen. That we've never kind of all had everybody get the same antigen at the same time before, where we know what's going on exactly, and we have like crazy good immunology techniques, and so we're able to learn like all these things that are sort of blowing our minds. And the place where my mind has been blown the most often is about the germinal center reaction throughout this pandemic. Brenna, yes.
I'm more interested in like the mechanism, because I thought we said that the germinal center would increase the maturation, but if you have the lower maturation, I thought we said the cells just kind of the antibodies die. So how did we get the broadening?
So I think that's something that people would love to know the answer to. And so what you might imagine is how far down the rabbit hole do I want to go? As the virus changes, some parts change and some parts stay the same.
Like the epitopes, right?
Yeah. So like there are some epitopes that are going to change a lot, while other epitopes don't really change.
Mm-hmm.
And that's kind of because of like which biochemical structures are like, can you change and which ones can you not change?
Mm-hmm.
Like I cannot easily make myself into an organism with 3 arms. That would be a pretty large change that would kind of destroy my whole structure. But I can make myself into an organism with blonde hair pretty easily, right? Like there are some things that like some changes that me as an organism or me as a protein can tolerate easily and still be me, and some that like I just can't. You can't do. And it seems like you might have, as this happens, more different viruses ending up in the FDC, and so the B cells might get better signal from the things that are the same across all of them, like having 2 arms, as opposed to the things that are different. And so you might get selection for things that are able to bind lots of related things. The number of controversial and difficult papers and pieces of immunology that I have like just like maneuvered around verbally is kind of intense. And so I could talk about this for many hours more and involve many more papers, but that's as far as I'm going to go right now. But there's much that could be said. Yes?
Just clarifying,
Mhm.
the broadening can only really happen if the germinal center is still active when you get reinfected?
So it seems like it's the broadening still going to happen. The other thing that we kind of think is that there is a similarity amount. So it's like SARS-CoV-2 antibodies can broaden to hit other SARS-CoV-2 antibodies, but they're probably not going to broaden so much they also hit influenza. So like, it's not like they're going to super jump to other pathogens.
Does that only happen during, like, after the first infection? So instead of, like, getting reinfected and then that's when it's happening? Or we just don't know?
I think there is a paper from the BNash lab that shows it after the first, but I don't remember, honestly. It's been a while since I've looked at those papers in that level of detail. Were there other questions? Yes, David.
I'm not sure if we talked about this the other day, but are journal centers like responsive to only one antigen, or like are they kind of like very?
The paper on that is from 2023. That really looks at that. And I said that you can imagine it being one antigen, and so you could imagine each of these might be, this might be one antigen, this might be one antigen, this might be one antigen. I said to imagine it that way. I can give you the paper if you want, because it's not super clear. But yeah, like I said, the point I wanted to make is how much the pandemic has actually really helped us learn about some of these things. So now, switching gears entirely, we're going to start thinking about a slightly different topic. And in thinking about this topic, I want to go back for a second and think about innate versus adaptive immunity. And what kinds of things define something as part of an innate immune response or an adaptive immune response. So if you notice here, we list the innate immune response as being something that happens really quickly, sort of within hours, while maybe we need a week for the adaptive immune response. And you've seen a lot of that going on here. With the innate immune response, we're kind of responding broadly to types of pathogens. While with adaptive immunity, we're getting that really specific response to a specific epitope, on a specific antigen, on a specific pathogen. Notice how many times I said specific. And you can see that with the specificities at the bottom as well. You can also see that with innate immunity, we often think about it being constant over the course of the response, while adaptive immune responses are improving over the course of their response.
And primary and secondary isn't synonyms for innate and adaptive?
No. Primary and secondary are different parts of adaptive. So adaptive the first time is primary, adaptive the second time is secondary. Adaptive the third time is tertiary. Adaptive the fourth time is quaternary. So like, for example, at some point I will talk through this example of like, you know, I get a flu shot every year. So like sometime in my life whenever I first got the flu, I made a primary response, and then the next time I got the flu, I made a secondary. And I've been making like tertiary, quaternary, whatevers each year as I get new flu shots. So those are all types of adaptive.
Oh.
The idea is that each of those times I'm making a new, better adaptive response.
Oh.
I am still making an innate response, but it's just the same old thing over and over and over
So
again.
it's more when we talk about the primary and secondary, it's like how many, the second time you were infected.
Yeah, and because the idea is the adaptive response adapts, it improves over time. And so kind of, at what level, how good is it?
Good clarification.
No, excellent question. You can see a couple of other things that people talk about in terms of adaptive immunity and defining an adaptive immune response here. And so what I really want to mention is, again, you can see that adaptive immunity, that ability in answering Brenna's question to adapt, that ability to have memory and change and get better over time, is one of the ways we define an adaptive immune response. That sort of expansion and contraction kinetics is sort of how we define an adaptive immune response. There are kind of two other things that I often think about as being part of this innate versus adaptive immunity thing. And so if I discovered a new cell, and I was trying to say, is this cell an innate or an adaptive cell? The two other things that I would think about are, does this cell do VDJ recombination? And so, where does VDJ recombination fit in to this definition? Yep. Oh, that was an itch, not a hand. Sorry. Where does VDJ recombination go on this slide? Yeah, Josh. Adaptive. So basically, the cells we learned about that are adaptive immune cells, B cells and T cells, did VDJ recombination. And all the cells we learned about that were innate immune cells did not do VDJ recombination. So you could be like, VDJ recombination could be the definition of what the difference is if we discover some new cell type. There's one other thing that we could think about as part of our definition based on what we've talked about so far this semester. And that goes back to hematopoiesis, the development of the cells. So remember that we talked about kind of all of these cells, all the white blood cells start out as bone marrow stem cells, and then they kind of branch in different ways in their development. We talked about two branches, the myeloid branch and the lymphoid branch. And so basically the idea here is you start with a hematopoietic stem cell. That hematopoietic stem cell makes a choice and either becomes a myeloid stem cell or a lymphoid stem cell. And then the myeloid cell becomes neutrophils and eosinophils and macrophages and basophils and dendritic cells.
Leukocytes.
Hmm?
Leukocytes?
Everybody's leukocytes.
Oh.
Leukocytes are white blood cells.
Oh.
If we come from a lymphoid lineage, we get a B cell or a T cell. We get a lymphocyte. So remember, we saw this way back at the beginning, right? So how does this tie into our definition of innate versus adaptive immunity? Yeah, Brooke.
Lymphoid is an adaptive and myeloid is an innate.
Yeah, so you could be like, well, if we came from the myeloid branch, if we came from the myeloid lineage, the myeloid stem cell, we must be an innate immune cell. And if we came from a lymphoid cell, we must be an adaptive immune cell. So yeah, we agree with all of this? So now I want to introduce you to a new cell type called an NK cell.
I couldn't even get the first two.
And this cell is called a natural killer cell. That's what NK cell stands for.
Sorry.
Natural killer cells are specifically, were specifically designed, said first or designated first as cells that were, that had this protein on their surface called NK1.1. That's not a perfect definition. It turns out that there are some other cells that also have NK1.1, which are a kind of T cell that I skipped earlier. I'm not going to care about those T cells right now. There are a group of cells called NKT cells, because people were confused, were they NK cells or were they T cells for a while? Now we know they're T cells. So NK cells have this protein NK1.1 on their surface. But the other thing that I want you to notice here in this slide, and you will in fact see this on another slide later as well, is that these NK cells, which are in this like pinky purple over here, are listed as a type of cytotoxic cell, similar to a CD8 T cell. And I will show you some other similarities with CD8 T cells in a second. When we look at NK cells, And we think about the development of NK cells, sort of where they come from in terms of hematopoiesis. You can see that NK cells come from the lymphoid lineage. So they come down the lymphoid branch. So they're on this lymphoid side. Here's the NK cell, which is also where the B cells and T cells are. If you think back to lab 1, where you were looking at these cells under a microscope, at blood cells under a microscope, this is what an NK cell basically would have looked like. You would have actually called it a lymphocyte. It looks under the microscope just like a lymphocyte. And so you probably may have seen some NK cells but not known it and called them lymphocytes. Based on this slide, is the NK cell an adaptive immune cell or an innate immune cell? Adaptive. Why is it adaptive, Brooke?
Because it's a part of the lymphoid.
Because it's part of the lymphoid lineage. So you're like, adaptive. Got it. Right? Huh?
Does it not do VDJ?
It doesn't do VJ. Among other things.
Dang.
Yeah, so
There goes my definition.
you see why I set you up with this definition, right? So, the next thing I want to tell you about NK cells is the timing of an NK cell response. So, in yellow, we're looking at the timing of a virus infection. Okay? In green, you're looking at cytokines that are made by the innate immune response. So you can think of the green as kind of being innate immune cytokines. The red is a T cell response. The blue is when we see good NK responses to this virus. And you can see where that falls time-wise. Based on this slide, are NK cells adaptive or innate cells? What?
Innate.
Innate. Why are they innate?
There. Almost immediate.
They're acting really early, almost immediately following infection. This is annoying, right? We got 1 vote for innate and 1 vote for adaptive so far. If I look at what NK cells do, if I look at the function of a natural killer cell, it is in the name. It is, in fact, going to kill things. And so you can see my NK cell killing either cells that are injured or infected by a virus pretty specifically. And so you can see that specific killing here on this slide where the NK cell is going to actually interact with the cell and lead to apoptosis of that cell. It's going to interact with that cell and lead to apoptosis of that cell, in fact, by providing some toxic granules to that cell that it's trying to kill, and you can see those toxic granules here. And if we even look further, I so almost love this slide. There's like one thing I hate about this slide and the rest of it's so good. Here's the toxic granules getting secreted. They should be secreted that direction. I don't know, this slide would be perfect if the granules came out that side. But they don't. They come out the wrong side. But here's the granules, and those granules are full of perforin, granzyme, and interferon gamma. Based on this slide, as well as the previous, what do you think about NK cells? Or what are you noticing about NK cells? Yeah, David.
Looks like they function pretty similar to cytotoxic T cells.
They remind you a lot of cytotoxic T cells, CTLs. Where are TTLs in this whole process? In innate or adaptive? Adaptive. So then kind of like, you feel like this should be kind of adaptive because it reminds you of an adaptive cell, right? So now we got some votes for innate and some votes for 2 adaptives and 1 innate vote. if we look at how NK cells get activated, which are the things I'm going to show you on the next 2 slides, I'm going to just say sort of briefly about them, and we'll start with activation a little more next time. One of the things that's really important for activating NK cells is type 1 interferon. So cytokines like type 1 interferon, which is made by the innate response, are really important in activating NK cells. That's part of the timing of NK cell activation. The other thing that's really important with NK cell activation that I will say so much more about on Monday are some receptors that are called NK cell receptors. NK cell receptors, as Brenna asked, and also as is true of the interferon receptor, has nothing to do with VDJ recombination. There is no VDJ recombination. They're just plain old genes that are plain old expressed, kind of like PRR genes. So in that case, innate or adaptive cells. Also, I will tell you that these NK cell receptors are not pathogen specific, and they don't bind to specific epitopes on a pathogen. They tend to actually be involved with very broad recognition that can happen with a lot of microbes. And again, we're going to say so much more about these receptors on Monday. So again, innate or adaptive? Huh? According to this, it's innate. And so now we have 2 votes for innate and 2 votes for adaptive. And so NK cells were one of the cell types that have helped us understand that in some ways our definition is a little wonky. And it turns out the immune system did not read the textbook and know that it was supposed to only be innate or adaptive. It didn't know that it wasn't supposed to be on the lines. How dare it? And so one of the things that's really cool about innate or NK cells is sort of where they fall in terms of innate versus adaptive immunity. In general, I always learned them as innate, but it always felt a little sketchy putting them in innate. Some data came out when I was in grad school that I will show you a little bit more on Monday that also made it even more complicated because that was also like an adaptive thing, and it was like, and so then the answer just became kind of their NK cells. And we actually now know of some other cells that are in the gray area as well. And so now there is just this sort of gray area between the two, but that's part of what we're going to talk about. So we'll talk a lot about these NK cell receptors, how NK cells work on Monday, and I will see you guys then. And make sure that you take a look at that paper before next Friday and answer those questions before the beginning of class on Friday.